Oh, hello all. My name is Kif Liakath-Ali from Stanford University, California. On behalf of the Bioprotocol Ambassador Program, it is my pleasure to welcome you all to our inaugural roundtable webinar. Today, I'm joined by fellow ambassadors, Dr. Matt Ogbu from Appalachian State University, North Carolina, and Dr. Swapna Naik from University of Connecticut, who will moderate the Q&A session of this webinar. The Q&A instruction is posted on the chat option. Please have a look. So the Bioprotocol Ambassador Program basically includes a community of early career scientists all around the globe who are passionate about reproducibility and to promote the discussions of the ideas of science communication, experimental reproducibility, open science, and use of preprints. A focus group or a work group within the Ambassador Initiative is dedicated to discussions on RNA-based technologies. So our first focus is on RNA sequencing and associated barriers in reproducibility and open access. When we were thinking about inviting a speaker to our first roundtable webinar, focusing on this particular aspect, we couldn't think anybody, none other than Dr. Ben Langmead, who is well known in creating a mega scale open access resources for the benefit of genomic studies. It is my great pleasure to host and introduce Ben today. Ben is an associate professor of computer science at Johns Hopkins University. He earned his Ph.D. in computer science from the University of Maryland in 2012. His group seeks to make high-throughput biological datasets easy for biomedical researchers to use. The group studies and applies ideas from sequence alignment, text indexing, statistics, and parallel programming. His group also has released several widely used software tools. For example, Bowtie and Bowtie 2. These are all very well-known tools in small read alignments in RNA-seq analysis and addressing common problems in genomics. His current work involves methods and software for efficient sequencing data analysis, methods and resources for making it easy to reuse and query archived sequencing datasets, and methods for alleviating bias and characterizing uncertainty in the read alignment problem. He is a recipient of Genome Biology Award 2010 and Sloan Research Fellowship 2014, a National Science Foundation Career Award 2014, and finally the Benjamin Franklin Award for Contribution to Open Access in 2016. Ben, thanks for agreeing to join us, and please take it away.
Great. Well, thank you very much for having me. It's my honor to be here, and I'm really glad to be in front of this group. I should be upfront about one thing today, which is that if you follow some of the links in this presentation, you'll find that unfortunately we are experiencing a hopefully brief downtime with some of the resources I'm talking about today. But we're working on that. I, in fact, have a meeting in about 2 hours, which I think hopefully will be an important step towards solving that. But at any rate, so I'm going to show you resources that are pictured on these slides, and the slides will have links, but some of the links you'll have to try again tomorrow to get them to work. This also another thing I'll say is I've sort of curtailed what I typically give as a sort of 50 to 55 minute talk, but I think this is okay. I've basically taken out some of the more historical and lower level details, and I'm going to try to concentrate today on what I think are some of the more compelling stories around how we can enable groups today to make regular use of already published public RNA sequencing data sets. Okay, so let me start with a figure that gives an impression of just how much of this sequencing data we have. Right, so we know that it's been a number of years that we've had second generation sequencing, and when people are producing RNA, people have been producing large RNA sequencing data sets, such that now our public archives, the most perhaps most well-known example being the Sequence Read Archive, which is stored at, there's one version in Bethesda in the US, but there's also versions in the UK and Japan. that this archive is growing very rapidly. And if you count up the number of bases of sequencing data stored in the sequence read archive, it's been growing exponentially. So this picture here shows the size of the archive over a number of calendar years going up to early 2022. And the vertical axis is on a logarithmic scale. It's the log of the total number of bases in all those data sets. And so each of those colored line segments, blue, red, is showing a doubling in the size of the sequence read archive. So the sequence read archive is an utterly enormous data set at this point. Very, very big. It's not the size of maybe the World Wide Web, but it's not too many orders of magnitude off from, you know, some of the most enormous data sets that computer scientists have to deal with. And yet we know that this is not all this is not a particularly easy to use resource, right? There's no such thing as Google for the sequence read archive. We can't immediately access and start using these data sets. So in one sense, it's, you know, smaller than the World Wide Web, but in another sense, it's also much less usable than some larger data sets like the World Wide Web. And so what our group has done, has, you know, sort of striven to do over the past 10 years or so, is to try to build large-scale systems that are able to process the data in the sequence read archive and turn them into more useful, usable data objects, so that people who want to be able to reuse public RNA sequencing data have a kind of nicer, cleaner place to start. They can get started much more easily. And so one of the strategies that we use to accomplish this is indexing. In fact, computer scientists, when we go to try to make very large data sets easier to use, we have a couple of sort of go-to strategies. One of them is indexing, and indexing is a big part of what I'll talk about today. There's a quote from George Bernard Shaw who says, "An index is a great leveler." If you would like to make a large data set usable, a good way to do that is to make it easy to jump to the part that you're most interested in, just like the index of a book. George Bernard Shaw did not say summaries are good too, but this is also true. So if the data set is too big for you to navigate in a sort of raw form, then a summary is a good way to get started, because a summary can take a large data set and turn it into something much smaller, but that you can make use of. And so all the strategies that I'll be talking about today employ some combination of summarization and indexing. This slide here is the sort of current progression of 3 projects that represent, in some sense, a software stack that enables something like a search engine for public RNA sequencing data sets. This bottom layer is about the pipeline that we use to analyze the RNA sequencing data sets in a uniform way. We have a tool called RailRNA, which is how we used to do it. There's actually a somewhat newer version that we call monorail, but it's related to this one. And that, I think I will not focus on today, but that is essentially the software pipeline that we use to produce these summaries, which are the foundation of everything that comes above. I will be talking very much about these top 2 layers, Recount 3 being our compendium of already computed summaries of RNA sequencing data sets, and then Snaptron up here being our question answering machine. So if you start with a more scientific question over splicing patterns, Snaptron is a layer that makes use of an index over the data that's in Recount 3 and allows you to answer questions rapidly with respect to those RNA sequencing data sets. So let me give you, starting from sort of this Recount 3 layer up, some more detail about what these resources are and how you can use them. And then I'm going to use the rest of my time to try to talk through a couple of examples where actual collaborators and scientists here at Johns Hopkins have used these resources to accelerate their research and make it so that instead of starting from designing a huge study where they do lots of sequencing, they start from the public data and use the public data to narrow their hypothesis space. So the first major release of this resource was back in 2017, and this version was called Recount 2. In Recount 2, we processed only human sequencing RNA sequencing data sets. So we went through what was then the sort of complete set of human RNA sequencing data set available in the sequence read archive. We found about 50,000 RNA-seq samples from the sequence read archive, and then from a couple of large human sequencing projects like TCGA, the Cancer Genome Atlas, and GTEx. We took, we had 10,000 samples each from each of these. And so in total, we had a compendium consisting of about 70,000 RNA-seq samples, sort of totaling to this size here. But it was human only. And for a number of years, this was sort of all we had available. And some of our colleagues were saying, you know what, we could really use, first of all, an update, more human samples, but then also several of our colleagues were quite interested in being able to study other organisms, especially mouse. So when we updated Recount more recently in 2021, when we published this paper here at the bottom, we did another survey where we took all the human and mouse bulk RNA sequencing data sets that we could find in the sequence read archive. We were able to update that 50,000 number to a much larger number for human, and then we were able to add quite a few mouse samples as well. And we brought along these TCGA samples, and since then GTEx had also been updated, so we were able to also increase the number of GTEx samples available in this resource. And altogether, the total amount of compressed input data that we processed to put into this new version of the resource was almost a petabyte, not quite. We were off by 8 terabytes. But anyway, it was quite a lot of data. And so the current Recount 3 resource contains study level summaries. And I will now summarize the different kinds of summaries that we have available, but it has study level summaries over all of those samples that I listed on the previous slide. So nearing many hundreds of thousands, nearing 1 million samples. So these are summaries, which is to say that even though our pipeline is capable of doing spliced alignment, which is the version of the alignment problem we have to solve when what we're studying is mature mRNAs from an RNA sequencing experiment. So even though our summarization does include solving the alignment problem, our summaries are at somewhat coarser levels of granularity. And this is by necessity because alignments are simply too big for us to store on a permanent basis for this many samples. So instead, we store downstream summaries that are substantially smaller than the alignments, but that nonetheless are sufficient for answering a lot of scientific questions. So we have summaries at the level of genes, so gene expression per gene in each sample. At the level of exons, gene expression per exon in each sample. At the level of individual splice junctions, so number of reads that span a splice junction in each sample. And then coverage level summaries. So for each sample, we have a coverage vector, right? So where it has, some positive number associated with all the bases that are covered by sequencing reads, RNA sequencing reads. So in other words, if the signal is high, that means that part of the genome was transcribed. So we have these 4 kinds of summaries, and we have them for every sample that I listed on the previous slide. And those are all available in the Recount 3 resource. If you were to navigate the Recount 3 resource through this interface, this is probably the first one to try. And so if you're going to write down one link from today, then maybe this is a good link to sort of get you started. What this interface does is it lets you search the study level metadata in all the studies that we've collected and put into the Recount 3 resource. And you can search for whatever you like. So in this example, I've put the term retinal here in this search box. And what happens down here is this view snaps, filters itself and snaps to just the studies that include the word retina in their abstract or title. And if you were to click one of these and then scroll down, on a normal day, you would see a set of links that allow you to download all the various levels of summaries that I just described. Today, it does not happen to be working at the moment, but it will be fixed soon, I promise. So this is a nice way to get started. And what you will be downloading will be a summary. So you might download a summary object where what you have is at the level of genes for each sample in that study, you have a gene expression value. Or if you choose to download the exon level summary, then you'll have for each exon and each sample in that study, value for how highly expressed that exon is. And these are, these are available at the level of tables that are text files. So you could get them as, for example, a typical delimited text file. That's one option. But another option that can be much more convenient is to get them as a summarized experiment object, which if you've used R and Bioconductor If you're not familiar with R and bioconductor, that's okay. But if you are familiar with R and bioconductor, this is a special kind of bioconductor object that's very useful because it's able to represent sort of matrix or tabular data like gene expression per sample, but in a form where all the metadata is sort of packaged into the object and permanently sort of stapled to it so that you do not lose track of either the sample level metadata labeling the columns or the row level metadata labeling the rows. So this would be a great place to start if you would like to get started with some public RNA sequencing data set summaries. What I'd like to talk about now, though, is this top layer, because this is the layer that helps us to get somewhat closer to the idea that this data is actually something where you can query. You can actually ask a question and get back a quick answer. And so Snaptron is the name that we gave to the layer of indexing and software that sits on top of Recount 3. It uses a collection of different indexing approaches tailored to the collection of different kinds of data that are in the Recount 3 resource. Some of those data are intervals, like, for example, exons and genes can be thought of as intervals with respect to the reference genome. Interval data is amenable to one kind of index data structure. And then some of the data is more numerical. This is amenable to certain other kinds of data structures. And then some of it is text, like the abstracts of the studies, for example. And that kind of data is more amenable to a full text index. So we have different indexing approaches that are combined in Snaptron. And when a user poses a query, the query might be concerned with all these different kinds of data. Like it might be asking, please tell me which are the gene, what are the gene expression values for the genes that are between here and here on the reference genome? And by the way, I'm only interested in the samples that have the word liver in the abstract, right? So it sort of combines all these different kinds of data, and Snaptron is able to take a question like that and break it down into a collection of queries against these indexes, and it's able to synthesize the answer and give it back over a web service query. So you can send a sort of typical, almost like a web query to this interface and get back an answer. So some examples of questions that you might ask using Snaptron would be, let's say you have a gene that you're very interested in. It's a gene that's implicated in a disease that you study. You might want to know for each of the junctions in the gene, in other words, for each of the potential introns that might be spliced out of that gene, how much support is there for that junction in all of the SRA samples? So in other words, which junctions are used frequently or which junctions are used infrequently? For a given junction, if you'd like to know whether it's an important one, you might ask whether its use seems to be correlated with something else. So for example, you can ask whether a junction is used in a tissue-specific manner in a data set like GTEx, where many tissues have been queried or assayed. And you might want to ask a question like, for example, maybe you have two splicing patterns in mind, maybe they're mutually exclusive with each other. You'd like to know whether the first or the second splicing pattern is particularly associated with some metadata, maybe a disease state or a cell type. And so you might ask, well, in which samples do I see splicing pattern A overrepresented relative to B? You can then examine that list and try to see if something interesting is coming up at the top. Okay, I think I mentioned already there are these two different interfaces, the REST interface, and then you can download the objects using R and bioconductor. Okay, I am now going to discuss a couple of use cases, and if you don't mind, I think I might skip this mini one that I've started with here. I will skip actually to the punchline, which is that we found a study wherein the authors were looking for novel exons, novel cassette exons, novel in the sense of not present in any gene annotation, in some data sets that they had obtained, and they found some, and then they validated them. And in our resource, if you were to use Snaptron to either ask the same question, where do I find novel cassette exons, you would have found almost all of the cassette exons that were identified in this study. In other words, the evidence for these cassette exons was there in the public data as expected, and that one way of discovering those novel exons would have been to query Snaptron. So I will, though, skip that one so that I can go on to a couple of more interesting ones that are more current. Okay, so this one was published in 2020, and it has to do with a particular kind of cell type called rod photoreceptors. You might remember, I'm not a biologist, but I did remember from maybe, I guess, high school biology that there are different types of cells in your eye that are used to understand different kinds of light, both colored light and light intensity, and they're called rods and cones. And so this story is about one of those two cell types, the rod photoreceptors, which detect light and transduce the signal to the brain. Degeneration of this cell type is an important cause of hereditary blindness in humans. And these cells are not particularly good at regenerating themselves in humans, so it's not very treatable. These cells are actually rather good at regenerating themselves in other species other than human, but in human, we're not so good at it for some reason. So the goal of these researchers here, Seth Blackshaw, who's a professor of neuroscience, and Jonathan Ling, who's a professor of pathology here at Johns Hopkins, their goal was to find splicing patterns that seem to be very specific to these rod photoreceptor cells, with the ultimate goal of seeing if they could create a kind of model system where they could create rod-like splicing patterns, but in a cell line, an unrelated cell line. Right? There's no cell lines derived from rods, but we do have other cell lines. So the goal was to take one of these other cell lines and see if we could induce rod-like splicing patterns in that cell line. So the organization of this study was, first of all, let's see if we can use public data to see, if rods and retinal cells have characteristic exon usage patterns. And for that question, we're going to look at both mouse and human data, purified tissue from mouse, GTEx for human. Then we'd like to know, are there certain exons that are only used? In other words, they're spliced in only in rods, and that would be using the mouse purified tissues because we can't really get the level of purification needed from the human samples in GTEx. And the third question was, can we find the splicing factors that are acting on and in some sense causing those splicing patterns of cassette exons used in rod photoreceptors by also asking which are the splicing factors that are overexpressed in rod photoreceptors. And then finally, can we take those same splicing factors but in a separate human-derived cell line and see if we can induce the rod-like splicing patterns to create something a little bit more like a model system, human model system for rod photoreceptors. Okay, so I'm going to walk you through 1, 2, 3, 4 in a series of pictures, and I'll go a little bit fast, but the main thing I want you to keep track of is the fact that as I talk through steps 1, 2, and 3, I am talking only about public data. Right, so that's the main point here. 1, 2, 3, that whole part of the study, which is most of it, is only looking at public RNA sequencing data sets. It's only when we get to step 4 that our collaborators finally do have to generate some of their own data. They have to do their own experiment to try to transfect these splicing patterns into a cell line and then sequence that cell line. But luckily, or not luckily, but maybe significantly, because it has implications for how other people perhaps are going to do their work in the future, this particular study, we only needed public data for the first part. Okay, so part 1 is just to show, this is a heat map. It's showing on these groupings of rows are different kinds of cell types, and these are all mouse data sets on this particular slide. So these are all cell types that were purified and then sequenced, RNA sequenced in those mice. And all the columns are different alternative exons. So cassette exons that might be spliced in or out, presumably in some cell type specific manner. And indeed, if you look at different groupings, and we're particularly interested in rods, which is this grouping right here, right, this set of rows that I've highlighted, we can see that pattern, the blue and white, where blue means spliced in and white means spliced out, is pretty specific to rods, right? That particular pattern is quite specific. So we seem to have found evidence that the splicing patterns for these cassette exons is indeed cell type specific. Interestingly, it is cell type specific in a way that we would not have necessarily seen if we had only looked at genes. Okay, this is a significant point, because a lot of what we do, especially with droplet-based single-cell data, is we count how many times a read falls in the 3' portion of a particular gene from a gene annotation. And, I want to sort of make you feel a little awkward and unsettled about that, because what we find, which is perhaps not really much of a surprise, is that the splicing pattern in many cases is way more cell type specific than the gene expression pattern. So here's an example of a gene sp10. If you look across all these various cell types that I was showing in the row space of the previous heat map, it has a certain distribution of expression level, right? It's clearly expressed most of the time, and its expression level moves up and down. But if you focus on one particular cassette exon and ask what fraction of the time is that cassette exon spliced in, this particular cassette exon, labeled here, is only spliced in a very specific cell type, which is cochlear hair cells, and it's almost 100% spliced in cochlear hair cells. Another example over here, but it's in this example, the specificity is to pyramidal neurons. So just one example of why it's a good idea, if you're interested in cell type specificity and very specific cell types, to look at purified bulk RNA sequencing data, because you get a very clear picture of what's going on with splicing. Okay, and indeed, these are just two anecdotes, but they're from a larger list of examples where we saw very cell type specific splicing inside of genes that were not particularly cell type specifically expressed. Okay, step 2 was we wanted to find these rod specific exons, in other words, exons that were spliced in only in rods. And so that's what this heat map is helping us to see, is yes, they exist, so that's good. Then we wanted to find what were the splicing factors that could influence that splicing pattern. And after some exploration, my colleague Jonathan focused in on these two splicing factors. These were actually partly known, right? So these were in the literature as affecting rod-specific splicing. And indeed, they were highly expressed in the rod cell types and not so highly expressed in other cell types. So this was a nice confirmatory result that these splicing factors do seem to be specific to rods. And so the final step was to take this same sort of cocktail of splicing factors and inject them into the unrelated cell line, which in this case was a cell line called HepG2, which is derived from, I believe, human liver. And what we found was that the splicing pattern induced by transfecting in some of these particular combinations of splicing factors, mainly these bottom two listed here, but this bottom one is probably the best one, does seem to recover some of these exons with arrows pointing to them that are spliced in rods. In human, we don't have rod level resolution, so we're backing up and looking at retina instead. But you can see that those splicing patterns that we identified in mouse, if we actually transfect them into this HepG2 cell line in human, they seem to induce the splicing in of these rods that seem to be, these exons that seem to be rod specific. And just jumping on top of that theme of cell type specificity being, of splicing being a good signal for cell type specificity, there's another layer of caution that I have to convey here, which is that we can't always rely on gene annotations to tell us what are the splicing patterns that we should be investigating, because the gene annotations are not complete. In fact, in this example, out of these cassette exons that I've highlighted here, 6 out of 8 of them are not actually included in some of the more inclusive gene annotations like GenCode, right? So if you were to just from scratch try to figure out, starting from a gene annotation, which of these exons are cell type specific, you would be missing the majority of the truly cell type specific ones, at least in this case. Which is perhaps not a surprise, right? Because gene annotations are based on, strong evidence obtained from bulk tissues. It's perhaps not surprising that therefore it's not very good at detecting the very, very cell type specific splicing patterns. Okay, so I'm going to wrap up soon, I promise. So this is the website where you can find this website should be working today, fortunately, where you can find the data from this ASCOT study. So human and mouse cassette exons, and showing you cell type specific splicing patterns for cassette exons in human and mouse. And then I want to finish with one more study. This one came out this year in September, just a couple of months ago, which is another example where our collaborators used public data to really good effect. And in this case, they were using it to design a very specific gene therapy. So in gene therapy, the idea is you want to deliver a kind of corrected gene to replace a defective gene, a gene that's sort of genetically defective, but you would like to deliver the corrected gene in the most specific manner possible to the affected cell type and hopefully to no other cell type. You want the correct gene to be expressed in exactly the situation where it's needed and perhaps no other situation, so as to minimize side effects. So the idea here is to design a package that gets delivered via AAV vehicle, so sort of like a standard gene therapy vehicle. But the idea is we're going to design this package so that the corrected gene is downstream of a cassette exon that may or may not be spliced in. And whether or not that cassette exon is spliced in affects whether the corrected gene is in frame or not. So the idea is if the exon is spliced in, the corrected gene is in frame and it's eligible to be transcribed and to do its thing. If the cassette exon is not spliced in, then the corrected gene is not in frame and there's no expression of the corrected gene. So our hope is that we can design this cassette exon and indeed this whole package so that the corrected gene is only expressed in the affected cell type and not in any other cell type. And this is called splicing linked expression design, or SLED. And so public data was used to try to pick which cassette exon should we put here, and not just the cassette exon, but the surrounding intronic sequence as well. It has to be selected. It has to be selected so as to not be too large, right? So one of the ideas is that in order to fit into this AAV vehicle, it has to be not so long. So it has to be less than 2 kb, the exon and the surrounding introns. And it has to be very specific to the cell type that we're actually trying to affect, right? So the idea is they used public data, again, same sort of data that I was just talking about, ReCount3, ASCOT. They used that public data to identify exons and surrounding introns that fit these criteria. And then they, showed that they had the desired effect in some models, right? So this is another application where, again, only public data was used to design these gene therapies. The only additional experimentation that was needed was to verify that they were working properly in those models. Okay, so both of those studies were examples where almost all the data used was public data, and then only the last few experiments required any new sequencing data, right? So that rod photoreceptor study, steps 1, 2, and 3, they used over 90,000 public data sets, and then step 4 was the only one where they needed to produce their own, and they therefore produced something like 8 RNA sequencing data sets in that whole study. They didn't need to do the sequencing to find the cell type specific splicing. That was done using public data. So our hope going forward, and maybe it's also the hope of some people here, is that the use of public data like this, of public sequencing data, becomes sort of an everyday activity. It's where a lot of projects start, as opposed to being something that's often either an afterthought or it's brought in only sort of toward the end as a validation step at the end of a study, a kind of in silico validation step. But rather to think of public data as something that can help generate hypotheses, narrow hypothesis space, and generally speaking, be a part of all the early steps of a project, all the steps of a project, not necessarily just the last one. And I have to end on a note of caution, which is to say that public data is not, this is not lovely, pristine stuff, right? Like, I don't know if anyone here has actually gone through the process of submitting data to the sequence read archive, but it's not an incredibly controlled process. People are not necessarily forced to use controlled vocabulary or to fill in all the metadata fields in a complete or accurate way. And it shows. And if you go to look at public sequencing data, you will very quickly arrive at instances where you're pretty sure that metadata can't be right, or the metadata seems to be very inconsistent with the data. And even if all the metadata were absolutely correct, there's always the problem of technical variables that creep in and that are especially important, that come especially to the fore as soon as you start to collect together disparate data sets that came from different labs, different labs that had ever so slightly different ways of handling the sequencing protocol, or that use different reagents, or that use slightly different maybe they use different read lengths. Maybe one of them did paired end sequencing and the other didn't, right? These technical variables become very important as soon as you start to pool data across several studies, because they can be the most important variables that describe the variability in the data. And so when we go from, maybe let's go to the public archive to extract one data set. Okay, maybe let's go to the public archive to extract 10 data sets. Okay, let's go to the public archive and try to look across all the data sets. These problems of incorrect or missing metadata and confounding due to technical variables gets bigger and bigger, and the sort of range of questions that we can successfully ask and answer maybe becomes narrower and narrower. And this is another very important direction for future research, is how can we address these technical variables, whether it's through better normalization, batch correction, things like this, so that it is more possible to look across many public data sets. Okay, with that, I want to thank all the collaborators on all these projects and yield it back to being a roundtable for the remaining time.
Well, thank you, Ben, for this fantastic talk. And it's really enlightening and useful resources. I mean, people like us who are biologists who do not have coding skills and computational power. I think this is great to look at a genome or splice form of interest to see where and when it is expressed. So I leave to the audience if they have any questions specifically. I mean, you can raise your hand and my moderators will help in addressing your question.
Yes, thank you, Dr. Langmead. That was a very interesting talk and very innovative. So before I head to the Q&A, I have a couple of questions. So at the beginning, you introduced and gave an insight about recount and snaptron, and you mentioned how the RNA-seq data is interpreted. You interpret based on the genes, exons, and junctions. So is there also a way that you can identify modifications in RNA sequences based on these? Because there are other RNA-seq data which do talk about RNA modification. I was just curious.
This is all through standard Illumina sequencing. I don't know, through standard bulk Illumina sequencing protocols. I mean, it may not all be Illumina, but it certainly is all short read, and it may not all be the same Illumina protocol, because I know, for example, there's a mix of some, poly A enrichment versus total RNA and things like that. But it's all variations on a theme, which is Illumina sequencing, which is, to my knowledge, is not really capable of reporting out things like RNA modifications. But I know, for example, that third-gen sequencing, when it's operating on native RNA, like a nanopore sequencing a native RNA, can potentially read out RNA modifications. Right. So maybe the only, this isn't what you meant by modification, but we can, in theory, see genetic variation in the alignments that we produce. We do not attempt to summarize that, in part because for some of the data sets we can't, or we're not supposed to, because of our data access agreement, like the GTEx and the TCGA data. We're not supposed to be distributing genotype information derived from that RNA sequencing data, so we certainly can't even do it there. But even for the other data sets, because I don't think we can do a very good job with detecting genetic variants. Therefore, we kind of, because it's RNA-seq data and we don't necessarily get, for example, equal representation from the two haplotypes and things like this. So even though we could, we don't make that available.
Okay, absolutely, thank you. I just have one more question from the, audience. So somebody said, hi Ben, Recount 3 looks like a powerful tool. Are you planning on expanding it to include more model organisms? And if so, which ones are on the top of your list?
So the Recount projects have been very collaborative. And, I would say from the beginning, my personal interest has mostly been in human, and then we expanded to mouse when it was clear that our collaborators who were ultimately interested in human splicing couldn't really do what they wanted to do with human data sets, because it wasn't really possible to purify cell type very well. And so they really needed mouse data sets, and that's the main reason we expanded to mouse. We could totally expand beyond human and mouse. Basically, any organism that has a good reference genome is eligible, right? And has splicing. It doesn't even have to have splicing, but it would certainly fit our tool set very well if it also had splicing. Where we do start to run into trouble, and to some degree we already run into it with mouse, is that humans are in some sense very well suited to having a single, well, there is, as was mentioned at the outset, one of my research focuses is bias that creeps in during the alignment process, and probably the most insidious form of that is reference bias, where we have one assembled human genome, maybe we have two now, that we can use as a reference. But wherever the donor individual differs genetically from that reference, it's harder to align those reads, and we get a little attenuation of the signal from the places that overlap the non-reference alleles. This becomes a huge problem if you're studying a species that tends to be inbred, right? And so some laboratory strains of mouse are quite inbred, and now the problem of, well, which reference genome did you align to? That question becomes very important, because if you pick the matching reference genome, you'll get less bias. If you pick the non-matching reference genome, you'll get more bias, more attenuation. And so you can imagine this is very true for like Arabidopsis and some species that that self or otherwise have inbreeding. And so luckily, we haven't really, well, I shouldn't say luckily, we haven't had to deal with that in a fulsome way yet. But if we expanded to more species, I think we would probably have to also tackle this question of, can we really get away with one reference genome, or do we need to start doing spliced alignment with respect to something more like a pan-genome representation for that species? And so that's one reason we haven't made that jump yet. But I would say I regularly talk to people who are very interested in us having more model species in there, and therefore it's entirely likely we will make the jump one day.
Thank you.
Thank you so much. Thank you so much, Ben. And thank you, my fellow panelists, Swapna, and the moderator, Kif. I also like to ask a follow-up question to what Swapna has asked. I think when you expand the tool, what kind of challenges do you anticipate to find or to get when you try to prepare the summary data that are often associated with the tool?
Yeah, I think the one I mentioned, that's the one that I'm most afraid of. I'm afraid that if we move to more species, that we will find, depending on the species, we will find more instances where what we're sequencing are pockets of relatively inbred organisms, such that if we don't pick the reference genome to match the genetics of that particular group of individuals that we've sequenced, that will end up with a bias that can in turn make it hard to detect splicing. For example, spliced aligners, like the one we use here is called STAR. Some of you may have used STAR. It's a popular spliced alignment tool. But STAR, part of how it decides whether a read really spans a splice junction or not is that it looks for those motifs. It looks for donor and acceptor motifs, and I don't think it looks inside the intron for like the branch site or things like that. It looks mainly at these donor and acceptor motifs. But if your reference genome has the donor and acceptor motifs in different places compared to the actual individual that you're sequencing, you can imagine the spliced aligner is completely confused. It has very little ability to get the right answer in terms of where the splice junctions are. So that's the main thing I'm afraid of. I'm afraid that the genetics of the underlying individuals will be a bad match for the reference genome, and that in turn will throw off the splice alignment process. I'll tell you what I'm not so worried about is the computational effort, because human and mouse are by far the most highly represented species in the sequence read archive. So expanding to other species, it will not take, the amount of computing power will be probably not even as much as we spent on one of the two species that we've already studied. And a lot of them have smaller genomes, which also helps make the alignment step, which is the most difficult computational step, helps make that step go quicker. So the good news is I don't think computationally it'll be a big challenge, but I do think that doesn't mean it won't be a big challenge. I think it will, but the challenge will be mostly in the bias and the interpretation because we will have to, on a case-by-case basis, on a species-by-species basis, really work to figure out whether the reference genome we picked is the right one. There's also the problem of the reference genomes being of not necessarily high quality as we move outside human and mouse, right? Human and mouse are some of the more curated reference genomes. As we get to other species, we might encounter some of the less curated reference genomes, and we have to worry a lot about whether the reference genomes are truly representative of who we sequenced. So that's what I would be most worried about.
When I asked the question, I had microorganisms in mind, considering their public health relevance and their abundance. But that's what I had in mind. At some point in my head, I was thinking probably would start focusing on certain region than the entirety of this species. What do you think?
Yeah, there's less. There's a lot less RNA sequencing data from, for example, bacteria, compared to what you might expect given the public health relevance. And I think that's mainly, someone can correct me. I think that's mainly due to the fact that the RNA sequencing protocols are pretty used to the idea that these transcripts are going to have poly-A tails that make them easy to grab onto with the RNA-seq sequencing protocol, and that this is not as true for bacteria. So I think it's partly that, but I do wonder, why isn't there? There really is very little RNA sequencing data in public archives from RNA and viruses compared to what we see for example, human and mouse. So I agree with you. I think that's kind of an untapped data type. But of course, we're limited by what's in the archive, right? We only know how to process what's in the archive, and right now the archive is pretty underrepresented in those areas.
I couldn't agree more with you, Ben. But also, considering the social value of this tool, which is enormous, what security measures, as well as what measures are you taking to, and you, by the way, when I mean you and your group, what measures are you guys taking to actually protect this tool? For instance, start off this webinar, you mentioned that the tool is down today.
Yes.
What measures are you taking to prevent disruptions like this?
Well, that's an incredibly good question, and we, as you can tell, we struggle with this question because I'll tell you, like, as a computer scientist, I definitely love building systems. I love putting together lots of different components of software and trying to scale them up and run them on bigger and bigger clusters. And but all the work that takes place in my group is in the context of PhD students doing their PhDs. And, I need PhD students to be able to work on projects that are going to be part of their thesis, right? And so, like, the construction of a resource like this is a huge project, and like being able to, this was a big project mainly for this PhD student right here, Chris Wilkes, who did a lot of the work, but also this Abby and Leo for sure also. But anyway, it was sort of the main PhD project for Chris. But then what happens when it's done? Now it's running, and you have to sort of keep it running. And so, and it's not the easiest thing in the world, given the way funding is typically structured, to hire and keep an engineer who would maintain a software resource like that. So one way that we've been trying to tackle this is that we work with other groups at Johns Hopkins who are kind of assembled specifically in order to keep projects like this going. At Johns Hopkins, there aren't many other genomics projects like this, but there are some other science projects like this, especially when it comes to astronomy. Like Johns Hopkins was, for example, the place that ran the Sloan Digital Sky Survey and did a lot of the sort of like control for the Hubble Space Telescope and such. And in fact, they're also the control center for the James Webb Telescope that we've been seeing the images from lately. And so there's actually groups of engineers over there who are quite experienced at maintaining databases like Recount, but for astronomy data. So we've been working with them, and we sort of are able to we have enough funding that we're able to sort of fund parts of those engineers, and we work together to try to keep the resource up and running with obviously some spotty success, but mostly pretty good success. But the question of how to keep these things running over truly long periods of time is pretty tricky, because it gets into the funding equation, it gets into the incentives of the university too, and for every engineer who works on a project like this, they need to have a manager, and they need to have people who are concerned about them and their careers, right? Like in other words, they're not just there for support, they're there to build their own careers. And I don't know how to answer your question besides to say it's really hard, and I don't think I've met anybody who's truly figured it out. But there are some really great examples of projects that do have really long and healthy lives. My mind immediately goes to the Galaxy project, which is maybe my favorite one to mention. That is a huge community. It's very much driven by and maintained by its own community, which is not something we've yet really been able to pull off with the Recount and the Snaptron projects. And so there are really great examples out there that I think do more than we've done on the community building side. And it could be that that's one area where we should improve. But I love your question, and I wish I had a really good answer to your question. I would say that in the case of what we've been doing here at Johns Hopkins, our best option so far has been to look around and find the other groups of people who have the most similar problem in terms of being able to maintain big systems over time, and work together with them and help fund them so that they can help us with our maintenance.
Kif, you raised your hands. Okay.
Yeah, thanks, Ben. I think that's a relevant question about this, how to keep these tools alive. And I often come across many tools that are dead. I mean, they start something as a big, and then it becomes useful. And as you mentioned, because of the logistics of funding and other stuff, they just can't keep it alive. And sometimes it's very frustrating that you cannot find the tool anymore, because a PhD students or postdoc leaves, or So another question that I had in mind, like equally as a bulk RNA-seq, right? So you also have the single-cell RNA-seq coming up these days, becoming cheaper. Everybody's doing, because everybody's doing. So can we leverage that to understand splicing?
Some of it. So like, for example, some single-cell sequencing protocols, not the most popular ones, but some of the protocols like SmartSeq are able to sequence whole transcripts. And in fact, part of the ASCOT resource that I mentioned is in addition to these bulk purified tissue samples, we also did a lot of SmartSeq single cell samples. But by and large, the popular single cell RNA sequencing assays are these droplet-based ones that sequence only the 3' extreme of the transcript. With a little bit of, because of random poly-T priming and stuff like that, a little bit of the interior of the gene as well, of the transcript as well, but mostly just the 3' end. And so those analyses usually are satisfied to end at the step of gene level quantification. They feel like they can't go any further because you can't really make out what's going on inside the gene. So some of the summaries that we provide are still quite relevant to even in that context, right? So for example, we have a gene level summary that we produce. So that should still be relevant for droplet based. But there are a couple other reasons why we haven't really moved into that space yet. One of them is that droplet-based single-cell RNA-seq is probably would require a special pipeline where we have to be very concerned with these barcodes and these UMIs that help us to distinguish, whether we're seeing essentially duplicate reads or not. That is not something we've tackled yet. But then the other part that I would say a little bit more positive statement as to why we haven't done that is because as far as we can tell, the people who have been most interested in using our resources are very interested in splicing, and therefore expanding to a huge number of cells where we actually can't distinguish splicing in those cells. It seems like it would be a hard move, and it would be into a space that's not really our advantage. But there are resources. There's really good resources that do exactly this, right? They start from a gene annotation, something that we avoid doing. But those other resources, they start from a gene annotation. They'll run a tool like Salmon or Callisto to do transcript level quantification, and they do it on all the public single cell RNA sequencing data sets. And one example of a resource like this is one called ArchS4, which I think still exists, and it was run out of Sinai in New York, Mount Sinai in New York. So there are some other projects that do that, but because we've been so interested in splicing, we've focused on things like bulk, SmartSeq, other data sets where we really can get the entire transcript and talk about splicing, which, as we argue here, if you're interested in cell type specificity, it's a little unsettling. The way the whole field has sort of moved in the direction of droplet-based single-cell RNA-seq, it's actually not clear what we're missing in terms of ability to distinguish cell types because of the fact that we're not assaying splicing when we're doing droplet-based single-cell RNA-seq. When we look at cell type specificity and what are the signals that highlight cell type specificity most clearly, they seem to be splicing most of the time. And so our collaborators are interested in staying in a world where they can see all the splicing.
Interesting. So, I mean, we have been using ASCAR for quite a long time since it's published, and we found it very useful. I mean, we did also have some validation in our own research group. So this is showing the collection or using the collection of RNA-seq data already published. So what if I wanted to identify a splice pattern of my gene of interest using an existing RNA-seq data in a completely different context, like a disease context or a knockout context? How can I use it? Like the way I would normally use ASCOT. Is it a...
Yeah, so if you have a gene, let's say you have, maybe suppose you're starting with a gene of interest because it's a gene implicated in a disease or something like this. Like I worked, for example, with a collaborator who's interested in type 2 diabetes. And so because of type 2 diabetes, he was interested in a specific gene, SLC30A8. I can still remember the name, even though I don't know very many gene names. And in that case, what he wanted to see was, well, please show me all the splicing that you've ever seen for this gene in all these public data sets. And I want to match it up with what we think we know based on the gene annotation and see if anything is perhaps missing from the gene annotation. And so we did this, and sure enough, there were some splice junctions that were missing from the annotation. And when we looked to see it in what data sets they were occurring, it turned out some of them were interesting because they were pancreatic cells, and so you could drill down that way. And so that's why we have the query type that I mentioned as sort of the third bullet when I was introducing Snaptron. I said one of our query types is, well, the first bullet was, you give me a gene, I'll just give you a picture of all the splicing I've seen for that gene, every splicing pattern I've seen inside the confines of that gene, which you could then cross-reference with an annotation. And in fact, we have a way of plotting them as sort of like arcs, where if they're annotated already, we plot them as arcs above the line, and if they're not annotated, we plot them as arcs below the line. So you can just quickly see which ones are annotated or not. Then another type of query is if you have in mind a splicing pattern that you think might be important for your disease condition, you can put in splicing pattern A, splicing pattern B, like maybe A is a splice in event for a cassette exon and splice B is a splice out. You can put them in and ask, when is A overrepresented relative to B, and you get a ranked list of all the samples, at which point you can start to examine the metadata of the samples. So if you're interested in disease state, you might quickly see or not see what the disease state that you're looking for in the abstracts and in the titles of the data sets that come to the top. So those are a couple ways that you can use Snaptron. Again, you don't have to download any data in order to do this. These are all just queries you can shoot off to a server. And this was actually how the ASCOT project started, is they were looking for patterns like this in the data that we'd already processed. But then they said, wait, you don't have any mouse. We really need mouse because we need cell type specificity, and we don't think we can get the purified cell types for humans. So can you please do this list? And so that was sort of how we moved from Snaptron to ASCOT. But it did start with our collaborators trying to phrase their questions with these sorts of queries with respect to Snaptron.
The Snaptron is not a web-based application, right?
It's a RESTful API. So it is, so it's hosted in much the same way that websites are hosted, over the same port and using the same protocol. But our expectation for how it's going to be used is either you use our command line interface, that's one way to use it, or we have a limited web GUI, which is the one that I was talking about that plots the arcs. But usually I think the way it's used is people use our R package called snap count, which allows you to frame queries and fire them off to the REST API and get your answers back. But if you wanted to, you could go to a lower level and just fire off the text queries using curl from the command line, for example, or other. So in other words, you can do it programmatically from Python or from the command line as well.
Thank you.
So on this note, we want to call the webinar over. And but while it's over here, we encourage everyone to join us on Slack, which would share a link to this Slack group right after the webinar. And also the recording of this webinar would also be posted in the Slack, so we can all keep the conversation on RNA splicing going on, and continue to learn and grow from each other.